So welcome to the engagement in a revolution that the world has never seen before, the genomic revolution. This is a time like no other. It's a time that we've, who have been in this field for some time, have been dreaming about. I really thought that this revolution would occur after my professional lifetime was over. I didn't think I would be so lucky to still be both ambulatory and hopefully cerebrally competent as this transformation is occurring. But this is truly an epic time in human history in which we are starting to recognize something that is as profound as the transition that occurred at the last century, which we learned at that point about the role of microbes and the importance that they play in our ecosystem and the concepts that they relate to as it pertains to health and disease. And if you think of the Pasteurian revolution that occurred at the turn of the last century and how it transformed healthcare and gave birth to sanitation, hygiene, and nutrition as being important parts of community health and built the public health system and ultimately caused the reduction of disease that far outstrips any kind of medicine that you can ever imagine. We are witnessing the same major kinds of transition today with the genomic revolution, and you've already been exposed to some of that front edge with the extraordinary presentations that we were graced with yesterday, and Dr. Lepine's presentation this morning. So Eric Topol at University of Scripps, basically, down at in San Diego, had challenged us recently in asking in this article that appeared in Cell, what if each of us knew our genetic uniqueness? Say that we were given that information when we were born. Would it change the trajectory of how we live over the course of life? And of course, the answer to that question is in part, well, what's the information content of that genomic information that we'd have? If it was just information that did no application to our lives, it might be just esoteric cocktail talk. Or it might be actually encoding a blueprint, which is the most precious thing that we own, which is called our book of life. Now think of that just for a moment, if you would. Book of life, what does that really mean? That means encoded in 23 chapters, half written by our biological father, the other by a biological mother, is not how we are going to be deterministically, but how we might be. It's a pluripotential manuscript that gives rise to all the ways that we can achieve our genius in life. And there's no human being that I've ever met that doesn't have somewhere locked into this magic book of life extraordinary capabilities that probably they don't even know about yet and are not fully tapped. So Topol wrote this book called The Patient Will See You Now, which, as you know, was his second kind of best-selling book that really was a very provocative, disruptive, innovative piece of literature. And what was the basic theme of that book? The title alone probably tells you, and that is, he said that we are moving into a period of time in which consumer activism, health consumer activism, will create a relationship in this virtual world of social media and internet information capabilities and the access to genomic and omic information in which the person will know more about themselves when they go to the doctor than the doctor will ever know. And therefore, the doctor will be lucky to see the patient. Follow what I'm saying? And so the patient will see you now turns this thing on its head as we move into this transition in the 21st century in healthcare. Now, you might say, is there a resistance to that concept in the favor-taking insurance reimbursement model for pathology-based histology in medicine? I would say, yes, there is a fairly significant resistance to that cultural change. And so we're witnessing that with people being naysayers and saying, well, you know, don't jump too quick, and we're still in control, we're the masters of the universe, and we have moved into an era in which we have democratization of information in a way never before, and that is empowering consumers to take charge. But with that said, as we have heard so eloquently expressed by previous presenters in this meeting, we want to make sure that the information those consumers have is authentic, is reproducible, it's been vetted properly, and it's not sending them out on a wild goose chase. It could either endanger their health or give them false information or lead to expectations that were going to lead to disillusionment. So we're looking for authentic ways of accessing this information. So where are we, and how do we find our way through the trees? And this is a really I think very, very interesting question, which is the foundation of this meeting, which has already brought all sorts of insights to you as it relates to some of the things that we know today that we didn't know, certainly didn't know when I started in this field in the early 70s, and really didn't even know recently within the last decade. So lots of accelerating new information. In fact, I've often said if you stop reading the literature for a week, you feel like you're intellectually stillborn. You know, it's so rapidly changing with a doubling time of less than 3 years now. Some people say 1.5 years. We accumulate as much new information in this field as we've accumulated historically in time going back to time zero. So it's a very, very dynamic, rapid accrual of information in which neuronal plasticity is required in order to keep up with the field. So where are some of the problem areas? And I'm going to give you some opinions, so please accept this as opinions and not necessarily always fact, agreed by everyone. But one of my opinions, which is a central feature about our Book of Life, is that we've recast it incorrectly. And we have put it into a context today that is now being owned by the histopathology-based disease diagnosis CPT code ICD-9 community. And why am I saying that? Because everything that we are talking about, and in this room, I think all of us are product of that culture, we think of it as genes for disease risk. Now think of that just for a moment. Once you introduce the term disease into your genes, you are forever locked into the same old model. The model is a disease-focused model. It's not a health-focused model. It's not a function-focused model. It is a disease-focused model. And you might say, well, that's really important because people respond well to fear. I don't think they do. Our political system is a good example of that today. So I think that, you know, people don't like to hang out in fear. And you might say, well, why do we have such resistance to behavior modification? Because people are fearful about what you've told them because of this some disease risk that they have in their genes, and they really didn't want to know about their ApoE4. And they really didn't want to know about their BRCA1 or 2, or they probably didn't even want to know about their detox SNPs because they feel there's nothing they can do about it. They've stolen their empowerment, and now they're in a situation of feeling like, I hope my number's not pulled out of the magic hat of disease. So is that really what our genome is all about? Risk factors? No, of course it's not. Our genes are quite different. There's joy in our genes. Our genes are there to express our function based upon the environment in which we live. Now, there is a life-changing, paradigm-shifting new concept upon which the new health economy can be built. Let me say it again. This is not a small idea. This is a major, disruptive, innovative idea that is now really what we're learning about our genome. Within that Book of Life, that double-stranded helix and the way that it's wrapped and folded and geometrically beautiful as the most perfect piece of jewelry ever designed, is the pluripotential that allows for, in the environment in which we select to live, starting in utero, maybe some we didn't select their in utero environment, but let's say however that occurred from that moment on, conception on, maybe even preconceptionally on, then is how that genome is going to be expressed into its function, which are really 4 basic kinds of function, as you know: physical function, physiological function, cognitive function, emotional or behavioral function. So our genes have the template to provide us opportunities to create our function based on how they're expressed. And their expression pattern is related to the environment in which they are being read. Now that is an extraordinary concept of plasticity, not determinism. Mendelian law that we learn is the most rigid genetic. You did the hybridized crossovers, you know, we're in the 8th grade, and you could do the pedigrees, and you can find out whether the peas would be fluffy coated or green or blue, or, you know, all those things of dominant and recessive, which are true, are really only part of the story of the variability that we possess within our genome based upon the environment in which we find ourselves. So yes, we have a coarse tuning knob called genetic recombination, and we have a fine-tuning knob called epigenetic environmental modulation, and those together give rise to who we are and how we look, act, and feel. Now that is a powerful concept that redefines the mystery of disease into the magic of genetic potential. Okay, so with that in mind, we might say, yes, there are genes now we're learning, the so-called single nucleotide polymorphisms, these single letter changes in the alphabet, that give us uniqueness among this variegated pluripotential that we have among the human kind. In fact, you probably recall that in the New England Journal of Medicine last year was a very interesting editorial in which the title was "The End of Race." The end of race. This is the New England Journal of Medicine. And what were they saying? You know, we've stratified clinical studies for years based upon this thing that we call race. You have to check on a box, right, as to what your race is. And now we find that their genetic variability within a race is much greater than the difference between races or among races. So what is race? It's just a social determinant for discrimination. So when we start thinking about the genomic paradigm, moving away from Mendelian determinism, which is what we all learn, it's encoded somehow epigenetically in our brains. In fact, we took tests to pass to go on to the next level in our scholarship, in which we had to recite that kind of catechism, which is wrong. Now, I'm not saying it's totally wrong. You get where I'm saying I'm exaggerating to make a point here. I mean, Mendel's laws still play a role, but I'm saying there's much more variability than we ever probably learned when we studied Mendel's laws. So many articles are now coming out in the peer-reviewed literature. I could, bore you to tears, go through all sorts of citations. I'm just doing a couple to kind of remind us, like this recent JAMA article, Genetic Variants Related to Serum Calcium and Coronary Artery Score, showing that there's a family of genes. In this case, you'll notice that they selected 6 SNPs that they could do prediction on the potential for calcium inclusion within the artery wall based upon these SNPs. And if we think that coronary calcium scores is a risk factor to heart disease, so this is kind of again, a risk disease genomic interrelationship. So I don't want to throw the baby out with the bathwater saying there's no value in risk assessment. That would be, again, another exaggeration. But I think this is a limiting concept, and I want to take beyond just the risk into kind of really what people want to know is, how do I function healthily for a century or more? That's really what people would like to know. How can I function healthily for a century or more? So that leads us to what Dr. Lepine was talking about. If we started at the place that is least challengeable, I think, in terms of genomic information, it would be in this pharmacogenomics area. Now, I want to recast slightly what Todd was saying in his talk, which I know he implied, but I want to make emphasis of it. So these specific family of genes that encode our detoxification potential, our pharmacogenomic potential to transfer drugs from a lipophilic state to a hydrophilic state so they can be excreted in the urine or the stool, those specific cytochrome P450s and those phase 2 conjugase enzymes that are encoded on genes, which represent a major portion of our human genome, our coding portion of our human genome, those are the same darn enzymes, the same genes that really regulate the conversion of all sorts of other things that are endogenous to the human body. We weren't given genes for drug detoxification because we didn't know about these drugs that we were going to be exposed to later in our evolution as a human. These have evolved. They're conservative families of genes that have autology that go down through all sorts of animals and plants, actually, which relate to the bioconversion of substances one to another so they can be moved through the organism and do their process and go out the other side and not accumulate, right? So if we didn't have that, think about your steroid hormones that you're making. I mean, if you didn't have these molecules that could make these steroids from fat soluble into water soluble, you'd turn into a big ball of cholesterol by the end of your life. So you have to find ways of converting in the form of cholesterol. One of the ways it's lost, probably a principal way, is through bile acids, right, which are water soluble. So you have your liver, which then makes bile from cholesterol. Now, I've just said something important there, haven't I? I've just said that these concepts of SNPs and genes are not universal to the whole body. They have organ-specific expression patterns. Now, let me say that again. When we talk about cytochrome P450s, where do you think of them most concentrated? You think of them most concentrated in the liver. But the liver has the same genes as does the kidney, as in the brain and the heart, so why is the liver doing CYP450s and not the skin? Well, because of epigenetics, right? In developmental biology, the genes that are associated with detox are silenced in some tissues by methylation, and they're active in other tissues like the liver. So if you start talking about the expression of the genome, you have to remember that these genes are not available in all tissues at the same level, at the same type. They're tissue-specific expression patterns. So when we talk about the intelligence of diet, if food is information, it's speaking to specific genomic patterns in specific tissues where these patterns are available for expression. Am I making sense here? It's not like a whole body effect. And this is an important clinical feature, I think, when we start talking about inducible genes. Aha, a new term I just threw out. What's inducible? Well, you have two kinds of genes, right? You've got housekeeping genes that are just doing the work every day, kind of carrying on the basic functions of the body. And then you've got these inducible genes that are responsive to environmental exposures that are turned on or turned off. And so the inducible genes, we might say, well, let's look at cytochrome P450s. If we look at CYP1A2, we already talked about its effects on caffeine. Well, caffeine then must be able to induce the expression of CYP1A2 in the liver. So if you're not drinking coffee, you know, your body's got the intelligence saying, well, why go to the problem of making CYP1A2? I don't really need it. So if you lived in a completely clean environment, you would have very low levels of activity, these inducible detox pharmacogenomic genes, as expressed in, when I say low level genes, I really mean low levels of the proteins that the genes make. The genes are going to stay the same, but the proteins they make are going to be not expressed very highly. If, however, you're in a less pure environment and you've got the genetic potential based on your SNPs, then those exposures activate those genes to produce those mRNAs that go on to make those proteins, and now you have that function. Am I making sense? So this is an important kind of variable to be considering when you're talking about, well, can I turn that on, can I turn that off, and if I can, in what tissue, and what are the activators that either are activators or inhibitors of that specific process? So it's a little bit more precision in the way we would think about nutrigenomics based on this model. Okay, so I look at my genetic detox profile, so that tells me my pluripotential. What are my SNPs that might not be so capable of being upregulated if they're exposed to certain activators? And so I have a whole bunch of homozygous SNPs for various detox enzymes that are downregulatable. In other words, they don't get the same stimulation. So as contrasted to Dr. Lapine, I probably need to be careful about certain things that he might have resistance to because of his ability to upregulate and to detoxify. So how does that relate to things like occupational exposure? And we think of, I'm going to take a social step over here into the troubled waters, I know, but let's look at this immigration situation that we're involved now with people coming from other countries and bringing other gene pools into our culture, so that we are a genetic sandpile in the United States, right? You go to Japan, you've got a fairly uniform genomic pool. Or you go to Iceland. That's a country, as you know, that's been used extensively for genomic testing. In the United States, however, we have a very remarkable sandpile genomic mixture. And so as we bring people into our country and they are going into certain occupations, they bring those genes into those occupations related to whatever those occupations might deliver to them as their environmental exposure. So I'm thinking of agricultural workers. So agricultural workers, as you know, have a lot of exposures historically to all sorts of things that we would say they're probably unintended studies in a long-term kind of interventional trial to see how their bodies respond to exposure to agricultural chemicals. Well, I could go on at some length about this and talk about chlorpyrifos, and I could talk about what's going on with Roundup, and I could talk about all sorts of things that are on the frontiers where people have rejected the anti-science nihilists of the world who don't know really how to tie their shoes correctly. Those individuals are still saying that this is all just based upon some kind of myth that we have these problems in our environment, and really we don't want to arrest business growth by having some controls on exposure. When these people know nothing about pharmacogenomics, they know nothing about relative risk, they don't even look at epidemiological literature, and probably they don't even care that there are populations at risk to fuel their coffers. So with that as a concept, I look at articles like this particular article that just appeared just a couple of weeks ago. The title of the paper is fairly self-evident: Occupational Exposure to Pesticides in Tobacco Fields, the Integrated Evaluation of Nutritional Intake and Susceptibility on Genomic and Epigenetic Instability. And what do they say in this abstract? They say that if you start looking at agricultural workers and their genomic sensitivity to exposure, you'll find that things like telomere length and epigenetic methylation patterns are different in those that have certain genetic predispositions that are then correlated with certain kinds of relative risks to agricultural chemicals that would be greater than others that don't have these genomic risks. So do we do pre-occupational screening? Of course we don't. And we know that if you look at, things like Parkinsonism, there is a long history. Glenn Stevenson and his work at Birmingham Medical School Neurology Department started back in the 70s to look at the prevalence of Parkinson's in occupations that are associated with, like in the paint industry, in the glue industry, various kinds of industrial solvents, that these industries have very higher levels of, in the semiconductor industry as well, of these problems related to exposure to these chemicals and neurological problems, because there's two sentinel parts of our body where the genes are most involved with sampling the outside environment. What are they? Neurotoxicity and immunotoxicity, right? And so where do we start to see clinically the most yellow canaries start to appear first? It's in the neurological area and it's in the immunological area. So it's autoimmune disease and it's all the family of neurodegenerative disorders. So these are important parts of what we're learning as we get deeper into this genomic database. So that takes me then to a moment of great respect. And I just inserted this slide this morning because I learned yesterday that I think we had failed to acknowledge an unbelievably important paper in this field yesterday. So I want to get it out there and make this the start of any study of genomic uniqueness to nutrition. And this is the paper that was published in 2002 by Bruce Ames and his colleagues, which is a landmark paper. And when I go back and think about the implications of this paper as a cornerstone to everything that we're now learning about nutrigenomics, it's a principle. So the title maybe is a little esoteric, but I'll try to describe it here. High-dose vitamin therapy stimulates variant enzymes with decreased coenzyme binding affinity, meaning the Michaelis constant, Km, relevance to genetic disease and polymorphisms. So yesterday, I don't believe we made a clear definition between mutations and SNPs, right? So let me try to make a clear definition difference, because you might say, well, aren't they the same thing? When you have a mutated gene, you have a letter change in your alphabet, so isn't that like a SNP? Well, the difference is in prevalency. It is true that at the molecular genetic level, they're the same, a SNP and a mutation. But SNPs are more prevalent. So when you get above 1% frequency of that single letter change in a population, we have arbitrarily, in the convention of the science, called that a SNP. If it's less than 1%, we call that a mutation, right? So inborn errors of infancy, metabolic diseases like Tay-Sachs, Gaucher's, Wilson's, phenylketonuria, various hemolytic problems, are all tied to frequencies in the population less than 1%. So those are called mutational disorders, monogenetic disorders. If, however, we have that appearing in the population at greater than 1%, and you saw that some of these SNPs are 30 or 40% of the population, now it's not a mutation, it's considered a SNP, right? So it's more of a definitional thing. Now, with regard to this paper, what is Dr. Ames bringing to our attention? And I think that the best way of me saying what he was getting across is, if you will beg with me, I'm going to read you this portion of his abstract, because I think it's beautifully written. As many as 1/3 of mutations, so you could call that SNPs, as many as 1/3 of mutations in a gene result in corresponding enzyme having an increased Michaelis constant. I'm going to do a parenthetical. What does that mean to a biochemist? It means that when that enzyme is in that altered form because the gene was a SNP, it's not binding to its active materials as it would in the native state, in what is called the wild-type state. So it alters its structure function, okay? The Michaelis constant is the way a biochemist would, or an enzymologist would define how tight that thing is bound together to make a reaction occur. So that decreased binding affinity for a coenzyme. Now, what is a coenzyme? This is a quick primer course and everything you already know about nutrigenomics. A coenzyme, you know, is the factor that binds to an enzyme that comes from a gene to make that enzyme active. So we have the the apoenzyme, which comes off the gene, but it's not really in an active form until it binds to its coenzyme, which is unique to its personality. And that coenzyme could be a mineral, like magnesium or iron, or it could be a vitamin-derived material, like thiamine pyrophosphate, flavonoadenine dinucleotide, nicotine adenine dinucleotide, pyridoxal phosphate, cobalamin. So these coenzymes bind to a specific enzyme, that was an apoenzyme, to make what's called a haloenzyme. The haloenzyme is the active form of the enzyme, okay? And if the gene is in a SNP form where it's less able to bind to the coenzyme, that means what? That it can't get its work to do as efficiently as it would if it was not in that form. I analogize this, and some of you have heard me say this over the years. I go back to my years as a baseball player in Little League and Pony League, in which, you know, among us boys in the league, the person that had the best glove was kind of like the king of the class, right? And so the way you made your glove really good is you put your baseball in there with linseed oil and you put it under the mattress and you slept on it, so that pocket was so good that no matter where the ball hit your glove, it would stick, right? And so this was very prized to have a mitt with a really good pocket in it. Well, that is the binding site for my enzyme, right? So the ball is the substrate, the mitt is the enzyme, and my hand in the mitt is the coenzyme, right? So if I got it all together right, then that ball, when it comes to me, it sticks in there and I get a good transaction. All the players give me a pat on the back. If I don't, then the ball bounces off. So there's one of two things I can do if the ball bounces off, right? One way is I can sleep on my mitt more. The other way is I can have my fellow players throw me the balls more often until finally I get enough to stick that it sounds like I really know how to catch. That's kinetic molecular theory, right? How do you do that then? That's called orthomolecular medicine. You keep throwing the ball to the mitt, which means you keep increasing the concentration of the coenzyme until you get the mitt to basically accept the substrate. Do you get where I'm going? Okay, so that's the underlying principle of nutritional therapy in the face of SNPs. Okay, so that's this paper. So I'm going to close here by just finishing out his abstract. He says about 50 human genetic diseases, you notice he hyphenated diseases there, diseases, due to defective enzymes can be, and when he say defective, what we really mean is they're SNP modified, so defective enzymes can be remediated by or ameliorated by the administration of high doses of the vitamin component of the corresponding coenzyme, which at least partially restores enzymatic activity. This is the foundational principle of everything you're learning here, right? In which the variant amino acid residues in these single nucleotide polymorphisms reduces coenzyme binding and enzymatic activity. They're likely then to be remediable by raising cellular concentrations of the cofactor through high-dose vitamin therapy. Okay, so we got that out now. That's a platform concept, right? That's an aha concept. That's a whole new view of how nutrients work beyond that of deficiency. So we're not talking about scurvy, beriberi, pellagra, xerothymia, rickets, squash, or marasmus. We're talking about functional outcomes based upon intermediary metabolism. So are there places that we can use this? Of course there are, and you're hearing all sorts of examples, like SNPs of the THF cycle and neurological function, of which we see many papers in this area. This is not like a mystery. You've already heard, many of these already being cited, so I don't need to go in great detail. So when I look at my genetic methylation profile, which is the folate cycle, having to do with S-adenosylmethionine, then I say, oh my word, I have multiple homozygous SNPs for some of these specific genes that are related to how that network regulates the availability in specific tissues of these methyl groups that are active for doing all sorts of things. So that would mean that maybe I would like that information just as I'm thinking about how I'm going to proceed through the world, how I might want my exposures, what types of diet I might want to be on. So it's starting to give me an interrogation. It's not an answer all to all questions, but it is opening up another door for inquiry to introduce me to the most intimate thing that I will ever have in my life, which is my book of life. When you introduce this concept to your clients or your patients, I hope that you introduce this as the most intimate inquiry they'll ever have to their most precious possession. It's not how they'll die, it's how they'll live. There is no more extraordinary opportunity to be introduced to yourself than your genome. It is the most privileged thing you'll ever have. And I think we're addressing this entirely wrong now, so it's a fear-based presentation, and the person then pulls back. They don't lean forward, they pull back because we've already set it up to be like, oh, you have some flaws. There is no such thing as a genetic flaw. There is no such thing as a genetic flaw. There's only genetic uniqueness. And in fact, there is a whole school of thought of which I would find myself as one, in which it is now recognized that what is considered a genetic flaw, if we look historically, might have been a survival capability for us as a species in our progenitors, in which maybe they survived an infectious organism because of a unique immunological thing that today, like the Pima Indians, if you use an example, living in this, people say, oh, the poor Pimas, they have diabetogenic genes. No, they don't. They have warrior genes. Just that we've now done a new experiment with the Pimas, putting them on the reservation and giving them white and see how it does. White flour, white sugar, white fat, and white alcohol. And now we see what the outcome is. Their genes are really selected for an environment of deprivation. And now we give them an environment of too much of too little. So genetic expression is regulated by this epigenome. I don't need to talk about that, except to say, look at the New England Journal of Medicine this year. I mean, you want to talk about the evolving frontier, the key role of epigenetics in human disease prevention and mitigation, right? This is where it's happening. I'm not making this stuff up. This is at the frontier of our advancing knowledge. Or The Lancet, a month later, Principles of DNA Methylation and Their Implications for Biology and Medicine. The whole nature of the epigenome is at the frontier of the top-tier journals in the world. And yet most doctors don't have a clue yet what this really means to them clinically and how it changes their transition of thinking from that of a histology, pathology-based model into a functional model. So we know about associations between global DNA methylation and telomere length in healthy adolescents, and we know that this is modifiable based upon lifestyle. Short telomeres can be found in people who live junky lifestyles, and they can lengthen their telomeres within 6 months if they do the right thing based upon their genetic opportunities. We know that plant phytochemicals are involved with epigenetic influence, so now we bring in this whole nature of signal transduction. It's not apoenzymes in the case of phytochemicals, it's modulating the expression of genes at transcription factors that regulate families of genes that are expressed to give rise to regulation of hormones and insulin signaling, inflammation, cellular replication. All of these are controlled by this magic relationship we have with secondary metabolites from plants. So when a plant is under stress, i.e. organic, it produces anti-stress compounds that when we eat them are anti-stress in our bodies. You want to talk a magic symphony that we're related to, that we've forgotten about? We think we have dominion over, multiply and subdue the earth when we are actually more a part of the earth than any other organism. In fact, the Neuraspis crassa, the red bread mold, probably doesn't care at all whether humans live or die. But we certainly care about the Neuraspis crassa because it's a fundamental part of our ecosystem that does all sorts of things. It degrades things that are waste into food that we eat, right? Really a powerful concept. So we know about all these methyl donors that are found not just from B vitamins, but found from phytochemicals, the resveratrol, the epigallocatechin gallate, even the hops phytochemicals that I've been working on over the last several years are known to modulate epigenetic regulation of these methylation patterns that then change in specific tissues the way those tissues express their genes. So that leads us from single genes to networks, and I'm going to close my thoughts to really just focus on really where we're going, I think. This is where we've been. Now I'm going to talk just briefly about where we're going. SNPs are wonderful, and you can see some really important utility of understanding these unique characteristics of these polymorphisms that have functional, I mean, there's several million SNPs, and not all of them are functional. In fact, the majority of them probably are fairly benign. But when you start to aggregate them together into families, now we start to recognize that there are patterns that we associate with network biology versus pathway biology. Remember, the way we learn and memorize and recited things was on pathways. That's really not how biology works. It's a time-dependent network that is emergent in its structure and function based upon the environment in which it's found. Wow, that's a whole different view of a dynamic biology than probably many of us learned. So I am colleagues with Joel Dudley and his colleagues at Mount Sinai and the Department of Translational Genomics. And he's been studying and published this paper looking at a disease, type 2 diabetes. Now, I think that the concept of disease, some of you know, is a fallacious concept. It's not that we don't have diseases, but we've misunderstood what they are. We think that these big placeholders like type 2 diabetes or coronary artery disease or systemic lupus erythematosus, that anyone that has that label is always the same as anyone else that has that label. And I'm saying that's absolutely ridiculous. Within any family that we call a disease is a variegation of all sorts of different ways that a pathway can lead to that person getting there, or a network. So what he has found in this study I'm just sharing with you is that there are 3 etiopathology subtypes of type 2 diabetes that require really have different trajectories towards different diseases, either cancer or to renal failure or to neurological disorders, that require different ways of approaching them. And so the concept of disease, now think of, if you would, ICD-9s came about after ICD-8s over about a 30-year period. Then ICD-10s came 3 years ago, and they had 2.5 times more diagnoses. Now, if we know about the primacy of disease, how do we suddenly have 2.5 times more diseases? Well, it's because we start to have cohort analysis of the diseases that we substratify, because we really didn't know what a disease was. Now we have ICD-11s just 3 years later, when it took 40 years to go from 9 to 10. Now ICD-11s have another 2-fold more conditions. What is that saying about our thought of the primacy of disease? It's a ridiculous concept that locks us into a form of wondering why we have so many outliers when that person had a disease that we could diagnose and we gave them the standard of care and it didn't work. Well, that's because they have their own condition related to their own uniqueness. It's the birthing of this precision medicine unit. So this is from this week's Nature Medicine. I thought it's perfectly applicable to what we're talking about. The title of this is GWAS to the people, right? Genome-wide association studies. You've already heard several of our presenters talk about these GWAS studies that have to have statistical significances of 10 to the minus 10th. You say, wow, 0.1 with 10 zeros, 0.10 zeros and 1, that's really significant. But when you take these GWASs and apply that gene to a human individual, it doesn't have much clinical relevance. That's what's made the pharmaceutical world so crazy, because they thought, oh, we're going to find the gene that really we can then pinpoint as a target for a drug, and we're going to change the course of trajectory and produce a whole family of new drugs. Well, it hasn't really worked out, because these genes are not expressed one at a time. They're expressed in families, and the families have small penetrations, but together they produce an aggregate effect. So what do they say? I'm quoting from this article. Thanks to improvement in data collection and analysis, some polygenic risk scores that predict disease are approaching the same predictive accuracy as monogenetic mutations. The time to consider incorporating polygenetic tests in the clinic is now. This is Nature Medicine, right? Polygenic, meaning not one gene at a time, not one SNP at a time. And so here are some examples of what I'm talking about. Prospects for using risk scores in polygenetic medicine. Now, we're linked into the Harvard Mass General group, our PLMI group, in which we have access, as you can get, to these new polygenic risk algorithms, which are the new computer scoring tools that have come out of really extensive research across 6 different common disease profiles that are looking at how you aggregate genes together and predict risk based upon the network biology rather than the single genes. And this paper that just appeared in Nature Review of Genetics about 2 months ago, entitled Genome-Wide Polygenic Scores for Common Disease, Identify Individuals with Risk Equivalent to Monogenic Mutations, is really giving now tools. These are real algorithms you can download and use that will allow you to pump or pipe your genetic information through these tools to do prediction based upon polygenic analysis. So now let me close with a classic example of how this all plays out. And if you've heard me speak over the last couple of years, you probably know this is a horse that I've been riding because I think it's so significant. There has been a question for years, ever since I started in this field, and that is, can you neutralize bad genes? In other words, if you got the bad luck of the draw and you didn't fill out an application card, is it woe is me and there's nothing I can do about it in terms of where my trajectory will take me. If my father has a heart disease, my uncle has heart disease, does that mean I'm fated to have heart disease? Or if my mother has breast cancer and my aunt has breast cancer, does that mean I'm going to get breast cancer? And there's this fatalistic view of genetics, isn't there? That we look at our family tree and we say, that's going to be me. So can you do anything to modify the expression of those characteristics that you might carry on your genes? So this study, which you know is Kathir's work and Kira at Harvard Mass General, is a really, really powerful work. This also, Paul Ritker is involved with this work as well, who you know is the HSCRP inflammation person. And so what they did, I'll just summarize this paper quickly. I'm not going to give it full credit, I'm sure. They took a cohort of over 35,000 people from 3 different clinical trials that have been done, 1 in Scandinavia and 2 in the United States, and they aggregated all this data together. These are people that had diet information, lifestyle information, genomic information, and they had followed them for a number of years after they have baseline data. And they asked the question, could we stratify or analyze those individuals based on their lifestyle and their genomic polygenic risk score into 3 categories. In the genes, we could say they're low risk, medium risk, or high risk based upon their polygenic gene scores that were built off GWAS data. And in their lifestyle, if they had a good lifestyle, medium lifestyle, or not so good, lousy lifestyle. So we would stratify them based on both of those characteristics. And if they, in their genes, had mono allele, we have 1 score, but if they had a double allele, homozygous, we give them a double score, right, for their genetic risk. So we weight this all together, and then we shoot it through the computer, and there are 55 different genes that come out of the GWAS for this risk to cardiovascular disease. And then they stood back and allowed the computer informatics to do its thing and asked the question, okay, what happens if you were in a high genetic risk group, the highest genetic risk group, but you had the best lifestyle? And the answer is, you probably know, that the incidence over a 5-year period of vascular disease was 50% reduced. 50%. Now, if I said that I could today stand up here and say I have a drug that I could take people at high risk and reduce their penetrance into disease, the most common disease, cardiovascular disease, by 50%, I would be wealthy enough that I'd take you all with me to Fiji. Yeah. I mean, that would be an unbelievable breakthrough, wouldn't it? But what have you heard in the public press about this? Virtually nothing, right? Virtually nothing. Now, the opposite is true, right? What about a person with a lower genetic risk with a lousy lifestyle? They had a higher penetrance, and the worst, obviously, was a high genetic risk and a poor lifestyle. So the point I'm trying to make is this genetic network concept of how genes and environment, lifestyle interface and interact is extraordinarily important. And you could break those genes down. I'm going to throw this out as a hypothesis. If you look at the polygenetic scores, to me, when I went through each of these genes, they broke into interesting families that relate to the etiology of cardiovascular disease. So they broke into inflammation genes, they broke into lipid metabolism genes, they broke into vascular function genes, they broke into vascular smooth muscle proliferation genes, and they broke into coagulopathies. So you actually can track some of the etiology based upon these families of genes that are associated then with these genetic risks, each one of which, if you think about it from a nutrigenomic perspective, you could start designing diets, couldn't you, for the individuals that have in-risk stratification in these areas. You could say, oh, gee, that person has a lot of coagulopathy genes. I need to really make sure I'm doing something to lower the stickiness of their blood cells, of their platelets. So polygenic risk scores is where we're heading. Bringing polygenetic risk scores to the clinic is the nature of the game today. We know that there is this thing called clonal hematopoiesis of indeterminate potential, in which our white blood cells, as we age, tend to be mutated, i.e. SNPs start forming and they get clonally produced, which then makes us not only have poor immune specificity, but most importantly, The genes that are commonly mutated are the TET genes, which are the 1011 translocase genes that are related to methylation and genomic epigenetic regulation of gene expression, which means we become more inflammation prone as we age. And if you look at people that are older, the level of their CHIPs, clonal hematopoiesis of indeterminate potential in their white blood cells, can be anything from 0 to as high as 30%. If you have a 30% chip level, you are at very high risk to virtually not just to hematological cancers, but to diseases like atherosclerosis, dementia, and arthritis. Now, is that modifiable? I would suggest yes, but it's only modifiable if you ask the right questions. Who has chips? And once you have them, can you turn them back the other way? And the answer is, this is the frontier of where we are in 2018. This is the epigenome methylation chip connection. We're starting to watch it evolve right under our feet. So what is the future? Here is my close. The future of nutrigenomics is an understanding of the relationship that specific nutrients have on an individual's polygenetic epigenetic connection to health and disease. As we unfold that and unpack that, we are going to have a revolution that will form for the first time ever a bifurcated system where we have disease care in hospitals and we have healthcare as a multi-billion-dollar industry that will stand at the table shoulder to shoulder with disease. And that is in our future view. Thank you very much.